Hi everyone. This is going to be continuing our lecture on DNA. Okay, so as we said before, A always pairs with T and G always pairs with C. And so because there's these specific base pairings, DNA can actually use that to their advantage in terms of how it replicates. And so what happens is in DNA replication, which happens during S phase, right, in interphase before the cell actually divides. When the DNA replicates, you have your two strands of DNA, the two parent strands, and then they separate. And after they separate, both of the parent strands can actually serve as a template for the new strand to be created. And so this parent strand right here will just base pair with all of its complementary pairings. So if it's like A, T, G, then it'll be T, it'll be all the complementary ones, and same with the bottom one. And so what you'll have is two new molecules of DNA that are exactly the same as the parent one. This model is called the semi-conservative model, and it's just a way that describes how DNA is replicated. And it means that in each new strand of DNA, you have one half of it that comes from the original parent molecule, and half of it is new. So one strand is called the parent strand, and one strand is called the daughter strand. So this is how DNA is replicated. So just to go a little bit more in depth with this, if you look at the original DNA molecule, you have the two strands that are like this. And there are multiple points called origins of replication where you can start to replicate the DNA. And so at these little specific points in the DNA, there's a protein that separates the two strands, kind of forces them to separate, right, and force away those hydrogen bonds. And then it forms like a little bubble. These little origins of replication form these little bubbles where the new strand is created on the insides of the two parent strands. And then it starts to expand these bubbles until they merge together. They kind of meet together, and then it completely separates the two parent strands and forms two new daughter molecules, with one strand coming from the parent and the second strand coming from the daughter cell, or is a new strand. So like we said, those points where the bubbles are created are called origins of replication. There are multiple origins of replication. So in one DNA strand, you can have multiple parts where it starts to create a bubble, and yes. Good. Okay, so there's something called the 5' and the 3' end of DNA. These are named after the number of the carbon on the 5-carbon sugar. And so here, for example, this point right here is the oxygen, which isn't drawn in, but here's the oxygen. The first carbon is attached to the nitrogenous base. There's a second carbon, and then the third carbon is the one that links to the next nucleotide. Then there's the fourth carbon, and then the fifth carbon is kind of like the one that goes off of the chain, off of the sugar, and it's directly attached to the phosphate. And so the 5' end of the DNA strand is the one where the 5' carbon, the one that's directly attached to the phosphate that shoots off of the 5-carbon sugar. This one right here is the 5' end, and the 3' end is just kind of when that sugar is flipped upside down, and the end has the 3', which is the one attached to the hydroxyl, is pointing up. So DNA is named after the 5', 3' strand. They run in anti-parallel directions, which means they just go in opposite directions. And in DNA replication, you have these enzymes called DNA polymerases. And so these are the enzymes that go in and add DNA nucleotides to the new daughter strand. Now, these DNA polymerases can only add nucleotides to the 3' end. So what that means is a new DNA strand can only grow from 5' to 3'. So just to make that a little bit more clear, if you take a look at this picture, here's the original DNA parent strands. You have the 5' to 3', and then you have the 3' to 5'. So when you separate the two parent strands, you're going to be building the daughter strands on the insides, right? DNA polymerase can only add from the 3' end, so it always goes from 5' to 3'. So you have one strand where it can just be built continuously. You start from 5' and you keep on adding to the 3' end, and you can keep on adding nucleotides. There's a segment of the other strand of DNA can't do that because it's not going in quite the right direction. And so what will happen is the DNA polymerase will create these short little fragments called Okazaki fragments. And they will build short fragments that go from 5' to 3'. And so on this bottom strand right here, it'll create these short fragments from 5' to 3', and then it will go backwards and build another short fragment, 5' to 3', and then it'll go back and create another fragment. So as the parent strand is separating, on this bottom strand, as it separates more, the DNA polymerase goes ahead and then goes from 5' to 3'. So as we said, one of the strands goes continuously from 5' to 3'. The other one is built in these short fragments. These fragments are linked together by an enzyme called DNA ligase. DNA ligase is just an enzyme that merges these little fragments together. And just so that you know, DNA polymerase can also proofread and check to see if it makes mistakes as it's replicating and creating new DNA strands. Okay, so we've talked about genotypes and phenotypes, but how do we get from one to the other? How do we go from having the genotype, which is the DNA, and then actually having the physical appearance of an organism? There are two main parts of that. There's something called transcription, which is when DNA becomes RNA, and then there's a process called translation, which is when RNA goes outside of the nucleus and then is read and turned into proteins at the ribosomes. So this process of, and then afterwards those proteins are what leads to your actual physical appearance. So we're going to go more in depth with these two processes, transcription and translation. Here's just a physical diagram of it. You can see that transcription happens inside of the nucleus, where the DNA is read to create these little messenger RNAs, and the RNAs go outside of the nucleus, where they're read at the ribosome, and they become protein. They can be read and turned into proteins. So for the first part, transcription, we're going to go more in depth with this later, but just so you know, the process is somewhat similar to DNA replication. The DNA two parent strands are separated, and then on one parent strand, you have an RNA polymerase that adds nucleotides that match that are complementary to the parent strand. And so if you have right here like an A, then the RNA is going to have a U instead of a T. If it has a G here on the parent strand, then there would be a C on the RNA strand. So it's pretty much the same. It's just that instead of having thymine, you now have uracil. So A goes with U and G goes with C for DNA, for RNA. Okay, so what happens afterwards, after this process, the RNA strand is completed. Remember, it's only single strand. It's not double strand like DNA. After this one strand of RNA is read off of the DNA, it leaves the nucleus and goes into the cytosol, where it goes to a ribosome. So after the mRNA is created, after it's transcribed from the DNA, it goes into the cytosol, and it goes to a ribosome where it's read. And then here what happens is three nucleotide segments of the RNA are read and translated into an amino acid. And so these segments of three nucleotides, you could think of them as like three letters that create like a word, and these things are called codons. So these segments of RNA called codons translate directly into an amino acid. And these amino acids are linked together and create a polypeptide, which eventually forms a protein. So this is a process of translation. We're going to go more in depth with both of these, but this is kind of like the broad overview. Thanks so much for watching. Bye.